we will Dr. Antonio through the sections Q&A, and we will forward the most voted ones to the speaker. Dr. Antonio, when you're ready, you may start.
Okay, thank you very much for the GVANC group for the invitation. It's always a pleasure for me to be not here in some University of São Paulo, but in contact with this meeting organized by my first home of graduate school. I have very good memories of those years at ESALC. So I'm going to talk about new challenges for serial breeding in southern Brazil. What do we think that might come, and what is the challenges we're facing from now on? So a brief outline of my talk could be, I'll give you an introduction, then some challenges that we have in southern Brazil. I'm going to show you some novel tools that we're using to explore genetic variability and examples and more. So I was reading some different literature for this, preparing this talk, and I came across this book I recently bought in Australia. And it says 50 plants that changed the course of history. And I was thinking, well, maybe even as though it is a great book, but it could be called 50 plants breeders used to change the course of history. That's the way I see how we are changing plants for the last 10,000 years, sometimes unconsciously now with more and more use of science. And these plants that are being changed by breeders are changing the course of history. We have some very large challenges to overcome. The world population is growing up, and it's expected to reach between 9 to 10 billion people in 2050. And that's something that concerns everybody, but especially the food production chain. And we know that the resources and the outputs and the inputs and the technologies are giving its share, but there's a big pressure to come from the genetics of these plants. So breeders will have a big challenge to change the slope of this growing trends in yield and reach all these empty stomachs. On top of that, we are currently facing more and more challenges in terms of biotic and abiotic stresses. Climate changes leading to flood, to drought, and new strains of fungi and insects. And also this population growth is putting stress on agricultural lands and pushing cities more and more into the areas of agriculture, forcing agriculture to move towards not so appropriate lands with more stresses to face. The classic plant breeding classes taught us that the ease we do breeding depends on the amount of gain we already obtained. Professor Ernesto Parteniani was my professor of plant breeding at the time I was at ESALC, he used to say that any breeder can do improvement. It's really hard to do to devolve this improvement, or to do a bad improvement of plants. The real thing is to be efficient. And how to be efficient if we are reaching plateaus in terms of genetic variability. So we need to be creative in order to find novel sources of variability to generate the new combinations that are needed to overcome the plateaus we have right now. So there are plenty of reviews that are warning ourselves that yield trends are not sufficient to meet the challenges of food needs by 2050. So we need to change the slope and increase the slope of genetic gain in order to achieve that. Here we have, and you have two lines. The full line or continuous line is the projected, and the dotted line is the required yields. And we see that for most of the 4 major crops, 3 cereals and 1 legume, maize, rice, wheat, and soybean, we are falling behind with the projected gains and the required gains. So we need to be more efficient in the next years. And what are the major challenges for plant breeders? I'll give you examples in rice and wheat. There are 2 major cereals. Probably the next speaker will talk about a lot about maize. So I figured that if I covered rice and wheat, it would be in a good complementation. So there's this picture from the Bill and Melinda Gates Foundation showing the 5 major challenges for plant breeders in rice. So we have submergence tolerance, which is a very common problem in Southeast Asia. Due to monsoons, we have flash floods, what it's called. That's our floods that increase very quickly and the plants remain submerged. When we have the return to the normal condition, plants, they die, they can hold themselves and die. So this has been solved with a gene called SUB1, which is a major gene that controls the growth of rice underwater and is able to reduce the effects of this flood on the yields. Other problems are related to drought, with yield losses up to 1.5 tons, salt to up to 2 tons, cold in some areas, and iron toxicity. So there are quite a few challenges for rice breeders all around the world to overcome these abiotic problems. In wheat, I selected a couple of biotic stresses that are important in wheat. One on the left is Fusarium head blight, which is a fungus that produces not only affects yield of wheat, it affects the quality of the flour because it produces mycotoxins. And these mycotoxins are harmful to the humans and animals that feed on this wheat. And we are now facing more strict regulations on the levels of these mycotoxins in the wheat flour. And we need to work better the types of resistance and the possibility of having more resistant plants in these areas. Another major problem that used to be restricted to the Parana state in South America is the blast. So blast, which is a common disease of rice, pericularia, the fungus pericularia, it was restricted to the Parana state, and it had very special conditions where it could grow and become a problem. But since 2016, there was some movement of grains from Brazil to Bangladesh, and seeds were supposed to be, the grains were supposed to be eaten, but somehow they ended in some farm fields, and now we have wheat blast in Bangladesh. And it's a very, very dangerous situation because Bangladesh borders with China and India, which are the two major wheat producers in the world. So we are developing projects involving these two biotic stresses, and I'm going to mention some of our work in a few slides. Another two important problems in wheat are harvesting sprouts, which is a problem in Rio Grande do Sul, where we have rains during the maturation time of wheat, and seeds germinate on the spike and decrease, of course, the quality of the grain. And acid soils, which are still a problem in major areas in South America, Africa, and Asia, South and North America also. Okay, so what is the southern region? We are south of São Paulo. As a joke, I come from very, very south in Brazil. I would say Pelotas is here. It's like 200 kilometers from the southernmost part of Brazil, which is Chuí. And I remember having fun with my colleagues in Piracicaba, where they would call people from Parana and Santa Catarina from the south. I said, no, they're not from the south, they're from the north, because I'm from the south. But it ends up that we are all in the southern region, we are all from the south. And we are responsible for over 80% of the wheat produced in Brazil and over 80% of rice produced in Brazil. So we have to face these major problems to help our region to continue feeding the whole country and exporting. The major stresses that we have in the southern region most of them are the same as globally. We have iron toxicity, we have cold, because we are so in such a subtropical area. We have colder temperatures in February that could affect the flowering of rice. So we have to deal with that. We have salinity problems. We have here is the major Laguna dos Patos. It's a major lagoon, and there's another one here, sorry. And there's a communication between the two. And this one sometimes gets, depending on the winds, it gets salt from the sea to the lagoon. And the rice farmers that produce here in Camaquã and São Lourenço, for example, they pump this water, and they could be pumping salty water. And some of them did, and some of the soils have problems with salinity now. For this lagoon, there's a solution. There was a blocking dam, which blocks the salty water from coming from here to there. And drought. Since we are here on the top of one of the largest sweetwater aquifers, the aquiferum Guarani aquiferum, we tend to think that we'll never run out of water in here. If you go to this area here in southern Rio Grande do Sul, it's a very lowlandy area, very flat and very swampy, and you have lots of water. So it's not common for people to think that we are running out of water or going to be ever running out of water. But we have demand for water from the cities, and the water is becoming more and more scarce. So it has some advantage to deal with some drought-tolerant genotypes. If not completely drought-tolerant, maybe more water use efficient genotypes. So this would be a good outcome for these areas. And regarding wheat, so I told you about Fusarium head blight, which is a problem in both states, with Rio Grande do Sul and Paraná, which are the major wheat producers. Wheat blast, it's mainly in Paraná, the problem, and now all over the world. And we have a project with FAO on production of induction of mutants and investigation of wheat blast tolerance in wheat. Pre-harvesting sprout, which is in Rio Grande do Sul, and aluminum toxicity, which occurs in both states. Okay, so moving to the next part of my talk. I showed you a large challenge that we have to face in order to cope with the increasing demand for food. We have seen that the yield gains are not at the rate we would like to. So what can we do? So there are many strategies that would help plant breeding to improve yields. Some strategies could be getting genes from wild relatives. So there are potential genes for tolerance to biotic stresses and abiotic stresses in wild relatives that could be used. But in order to use efficiently this source coming from different germplasm, we need to be able to identify the genes that are interesting that can be transferred. So I'm gonna talk about genome integration and mining, wild relatives, some genome-wide association studies, and some meta-Qtl analysis. So for gene integrations from crop wild relatives, it's been now 15 years since we published the map-based sequence of the rice genome. We participate as the Brazilian member of this consortium that sequenced the rice genotype Nippon-bari. And when we did that, just after we finished the rice genome, and there was a brainstorming colloquium to identify, okay, what's next? We finished the rice genome, what do we need to do? Well, there were many different points of view. One of that was to find every function for every gene we sequenced, and we found 25,000 of them. And also there was an idea of starting an international horizon map alignment project. That was led by Dr. Rodwing, University of Arizona. And we jump on the board to do the next step that would be then to find the genes from these wild relatives, these wild genomes. So in 2018, we published the genomes of 13 domesticated and wild rice relatives, showing the turnover innovation across the genus Oryza. So this was an effort to, again, as the rice genome sequence was an effort to provide a great tool for breeders to find genes that were better suited to their varieties. This was also a step to find, to make the accessing of new genes more feasible. There was mostly this genome size range of between 261 and 394 megabases, and a range of genes between 2,000 and 36,000 genes. There were some genotypes that were left. See here, most of the diploid ones were used in this study, and the tetraploid ones are being further investigated. We collaborated in the sequence of Oryza glumepatula, which is a species endemic to Brazil and South America. Very important outcomes, identifying the different rearrangements and the genus and how this species evolved and which were the closest relatives to the cultivated Asian and African rice. And of course, one example that was shown in the paper was to study some disease-resistant genes and the families in different genus. We did similar studies on sub1 gene that was published in Rice. And also we have one paper submitted now looking at grain quality traits or starch producing genes. As you can see here, different rearrangements of these NLR genes in the Oryza, and there are a bunch of combinations of orientations and duplications of genes, which is expected because of the nature of disease-resistant gene is a way of plant coping with the diversity of the high variability of the pathogen is to duplicate genes and make tandem duplications and rearrangements in order to produce novel sources of resistance. So many resistant genes were identified in wild parents of Oryza. Some strong candidates, the source of PTA2, which confer resistance to blast. And with this new tools, we could identify new orthologs in the AA, BB, and FF genomes of Oryza. So this was a tool important for making further studies of transferring important genes into cultivated germplasm. Another project now in wheat, shifting gears to wheat, it's a collaboration between our group and the group of Dr. Ian and Julie King, Doctors Ian and Julie King in the University of Nottingham. And as a part of a partnership between CNPq and BBSRC, and we sent PhD students and postdoctoral fellows to Nottingham and did some integration into Brazilian germplasm of wheat. We don't need to see that. That's the production of wheat in Brazil. I already showed that importance. So here, the genetic variability of wheat is low, because if we look at the origin of hexaploid wheat, it's only between 8 and 10,000 years ago. So that's very recent, in order to accumulate high variability. There is, of course, an advantage of being an exploit where different genes can be shut down in different genomes through epigenetic factors, and then you can see the plasticity of wheat in terms of this epigenome. But even so, if we look for markers, wheat within the germplasm, elite germplasm of wheat, there is narrow variability. So there is this very, very wealth source of novel genes in the wild relatives of wheat, and this can be, if only we can figure a way of transferring genes from these different species, that would be a very good way to improve or increase genetic variability in wheat. So one of the projects that we were involved was the introgressions from Agilops peltoides and from Triticus timofeevi. This is a way how the scheme of how the wheat introgressions were done, crossing Agilops with wheat cultivars, then the F1 again, and then you have backcross 1, backcross 2, and then you start with this backcross 2 to the Brazilian, and this Tibio sinuelo is a variety produced by Biotrigo, which is a wheat breeding company, a private company situated in Passo Fundo, and transfer these segments into Tibio sinuelo and obtain, of course, plants with introgressions. Here you can see how the novel tools are important. So this fish studies, GISH and SNPs. We can look at the segments that have been introgressed and detect them using GISH. And you can see here where you have the different genomes, the A, B, and D, and then you can identify that this fragment is really introgressed into the a chromosome from the B genome. Then you can look at the recombination events and see which chromosomes have the larger introgressions, which ones have smaller introgressions. Preferentially, small introgressions we have the less linkage drag, the last effect of linkage drag, bringing some undesirable genes from these wild relatives. So we are looking for smaller integrations that are stable and that then showed some valuable phenotype. Here is also fun to see how the synteny can be compared between wheat and timofeevi. So you have the genes at 6A here and orthologs to parts of 1A, 1B, and 1D, and all sorts of comparisons can be done. Some of the chromosomes are very, very conserved, and some have translocations. That's the history of wheat. And this information can help us to identify which translocations could be useful for novel genotypes. So the results of the second part, we identified 600 segments in all generations, 316 in the Brazilian cultivars, all groups with transferred segments, and only one BC4 plant did not receive any segments, and many plants have received some single introgressions. These plants are being further studied now. We were planning to bring the seeds to Brazil. With this situation of COVID, we are now holding until the situation improves. The last part I'd like to mention is another tool that can be done with the information already available in literature, which is Qtl Meta-Analysis. So again, we took an important problem for wheat in southern Brazil, which is Fusarium head blight, and we looked for the results in the literature for Qtls conferring tolerance. This nasty mycotoxin is, in order to get rid of it, we need to understand better the resistance present in the wheat germplasm. It's best way and more environmental friendly and cost-effective way of controlling this disease. So we have different types. Is a quantitative trait governed by many genes, and it has been proposed that wheat has 5 types of Fhb resistance. Type 1 would be to initial infection, type 2 to the spread throughout the spike, type 3 to mycotoxin accumulation, type 4 to kernel infection, and type 5 to yield losses. Technically, they are different traits, but somehow they interact. And we can see some differences among the wheat germplasm on the showing of this resistance. A few years ago, we published in Journal Ciência Agrícola, we published a survey on Fhb in wheat cultivars, and we were able to show that some of the Embrapa cultivars like Parudo is one of the best in terms of accumulating trichotillins. You have DON, nivalenol, and nivalenol here, which DON is produced by Fusarium graminearum, which is a different species, and Fusarium meridionalis. Both are mycotoxins that are bad for health of the people, and you can see that there are some of the genotypes accumulate high levels of these mycotoxins. And when you look at Parudo, it's low and low. Also Frontana, although it's high here, is very low on this NIV. So this was an interesting way of starting to see if there was variability for this resistance and the Brazilian germplasm. Then, so we could point out the importance of this genotype for being parents for the next breeding generations, and also we decided to move on to the Qtl analysis. So Qtl mapping, which is a very important tool, has been around for more than 2 decades, and we were able to recover hundreds of Qtls described in literature. And how can you, because these are different populations, different biparental populations, different environments, so the Qtls express themselves in different regions of the genome. It's very hard to look at this paper separately and say, okay, let's focus on this region, genomic region, or in another genomic region. So if you want to use this information for marker selection, it would be nice to see, okay, let's see which Qtls remain. We compare and we compile all the information from different literature. So the aim of this study was to perform a comprehensive meta-analysis of Qtl related to Fusarium head blight resistance in hexaploid wheat and integrate genetic, genomic, and transcriptomic data. So the first questions were, is there any redundancy? All of them are all of them unique? Or if the Qtl already considered redundant could be actually novel, unique? Is there any possibility to refine the mapped loss in terms of length and see if we can mine the genes within those Qtl regions? So an important tool was the genome sequence that was released recently in Science from the Chinese spring cultivar. And we integrated genomes and transcriptome for identifying the candidate gene mining and their validation through the transcription profile under the Fusarium head blight within Qtls. And of course, we looked for functional validation of protein evidence for these genes. So there was, here I'm not gonna have time to go to all these steps, but there was a very organized step: collecting Qtl, searching maps, building a consensus map, organizing data, calculating the interval of confidence intervals, doing Qtl projections, meta-analysis, candidate gene mining, cross-validating with transcripts, searching for protein, and then have some candidate genes coming. So there were like 7 maps used for the consensus, using SSR, synthetic times Opata, some version 4 of which consensus map, some Illumina 9K, Illumina 90K, and some GBS map. And then we joined everything. And 556 Qtl were collected from literature, divided here in different chromosomes. So this is the subgenome A, subgenome B, subgenome D, and then the different chromosomes within the subgenomes, and seeing which were the stronger or the strongest occurrences within the DUE genome. Okay, looking at the markers, the consensus map have the length and the number of markers, and produced a very dense genetic map with 572 markers. And as a result of the meta-analysis, 359 Qtls were able to show 65 meta-Qtl, good refinement. He also, there was a refinement in the length of the lossy, narrowing the genomic regions that would make it easier to find genes. As you know, wheat is a very large genome with repetitive elements. You have large regions, it's hard to find all the genes that are present. I just show you one of the, so we have this for all chromosomes that showed some Qtls. And here are the types of Fhb resistance in colors, 1, 2, 3, 4, and then 1 and 2. And then they are colored here as different mapping. And chromosome 1a then is a good candidate. And then there was a projection of the collected Qtls and the analysis. So as a final consideration, and probably the largest study of this kind carried out on bread wheat, and the first to use genome annotation and transcriptome data jointly. Results obtained through this integrative study will help breeders to better use the available Fusarium head blight resistant genes in their breeding programs, as well as to contribute to the understanding of the genetic and genomic architecture of Fhb resistance in bread wheat. We have now two strategies we are pursuing. After this, we are doing some panel of wheat genotypes. About 300 genotypes will be sequenced in collaboration with CIMMYT. And also our idea is to develop cast markers from these more important regions for Fhb resistance. So to finish, I'll leave you with a message that says from an ancient Chinese proverb: if you are planning for a year, sow rice. If you are planning for a decade, plant trees. If you are planning for a lifetime, educate people. I have to thank Eduardo Vansky, which is a postdoc in my lab, which did the wheat meta-Qtl analysis. Railson Santos looked at the, and Daniel Farias, which was here. Not here anymore. And they did the RISA sequencing. And also Eduardo was in Nottingham. There was another 3 or 4 students in Nottingham. They are not here anymore. My group is composed of myself, Professor Luciano Maia, and Professor Camila Pegoraro. And we all share lab. Oh, here, Daniel Farias, the Rhiza glumepatula genome. And I'll be happy to answer any questions. Thank you.
Thank you, Dr. Antonio. I would like to thank you for your great lecture. It was a pleasure to hear a little bit about your research. I'm sure it was a very useful topic for all attendees. We have 3 questions here, and I'm gonna read it for you. The first is from Carla Gasparini. Is there any incompatibility between wheat species? How select the species to perform introgression? Are you hearing me well?
Yes, a little bit. It's not too high, the sound. But well, it's a matter of making crosses and getting good survivals. Of course, there are some embryo rescue techniques that help when you do this interspecific crosses. And the group in Nottingham maybe is one of the last very good in cytogenetics, and they have been doing crosses, lots of crosses for all these relatives, and it's amazing the amount of work and the amount of results they are getting with such a small group.
Okay. There's a second question from Jose. Hi, Professor Antonio, congrats for the great talk. Are there any association between the genes for tolerance to Fusarium with BLAST? About the translocation 2AS, 2NS, what is the level of tolerance that it confers to BLAST, and what are your thoughts about their association with quality and yield? I mean, do you believe that using wild genes could potentially affect yield and quality?
Well, the Fusarium and BLAST are different diseases. There could be some general response genes that respond to both, but I don't believe that get one. It would be great. It would be great to have one gene that fits all, that is both tolerant or resistant to Fusarium and BLAST. No, the Fusarium strategy is that I show you. We did first is Qtl. Now we are doing inoculations in the greenhouse, and doing phenotyping for over 300 genotypes. We have more than 400, but I think now we have 300 already phenotyped for Fusarium. And we have prepared lyophilized tissue to send to CIMMYT for genotype. So we will do some GWAS analysis for Fusarium head blight in the next 6 months or so. For BLAST, what we have is we are doing some, we are a little bit behind. We are doing some Qtloms or meta-Qtl analysis. And we also have mutants. They're EMS mutants that we generated for the mutation breeding, and we'll be starting to identify or to screen mutants for BLAST maybe next year. We have also, I have some students looking at the protocols for assaying BLAST, wheat BLAST, and because it's a tricky one. It's in Rio Grande do Sul, it's not really a very common disease. As I said before, in Paraná, there are the right conditions, but we are managing to have some responses, and there are some, those pictures I showed are from my students. So they are already succeeding in having inoculations and showing differences between resistant and susceptible genotype. This BLAST project, which is funded by FAO, FAO-IAA, it's really something that it's under a red light, let's say, because there has been some descriptions. I just got an email last week that wheat blast has been identified in Africa also now, and something that was up to 10 years ago was only described in South America and Paraná and Paraguay. Now it's being spread all over, and we don't have understanding of the fungus is similar to the rice one, but they are two subspecies, and we hope part of the project, and we have a CNPq grant, is to look at the comparison between the genes for resistance to blast from rice and wheat and see how similar they are, if they have orthologous loci, and what can we infer from that. I hope I answered the question.
Yes. So the next is, thanks, Professor, great presentation. Is the paper related with the meta-analysis of the Qtls for wheat already published?
Yes, in Frontiers in Plant Science. And the first author is Vansky. Sorry, I had it on my presentation. I was mentioned to Professor Baldin, but I had not these slides, and I thought it was too much. So I deleted some, and with that, I deleted the one showing the paper. But it's Frontiers in Plant Science, and was published October, November last year.
Okay. I have another question. Besides the breeding for resistance traits, do you have in mind any kind of breeding for improving nutraceuticals properties or biofortification? How do you see this trend in wheat breeding?
Yes, we have, and we have already going on this panels that we are doing. We also doing phenotyping for presence of elements, let's say iron, zinc, copper, and I think we have a paper already out on trace elements journal. yes, there's an interesting variation in the accumulation of these elements. And there's a whole story that I could talk more another hour about, but we started looking at arsenic because in rice, and then we looked and also other elements in wheat and oats. So we have a partnership with the University of ABC, Professor Bruno Batista, and he does the ICP-MS, and we are analyzing the contents of these different genotypes. And it's a great tool because then we associate with our genomic genotyping from IRI and from CIMMYT. So we have the rice and the wheat genotypes for association mapping and the phenotyping from the ICP-MS. So yes, we are also doing some biofortification studies.
Yes, okay. There's a last one from Emerson Limberger. After 15 years of experience with rice genome, what are the key learnings for studies with the recent wheat genome?
Wheat has a much different genome than rice. Wheat has a genome that's 40 times larger than rice. And what I think my personal opinion is that we will find more and more epigenetic influence in wheat, since there is such a wealth coming from these interactions between the different subgenomes. And so although we don't see much variability in the sequence, we see a lot of plasticity in the genome, and my opinion is that is due to epigenomic or epigenetic effects. We have a paper coming out that was a long time ago, but we still have the results unpublished. Was the survey of 22 megabases on the wheat Chinese spring, looking and comparison of the wheat landscape in terms of gene-rich regions and gene-dense regions. And it looks like that we had wheat would be, let's say, have smaller islands, more with gene-rich regions sparse through the genome, other than have one big island of genes and very large stretches of repetitive DNA. But that will come soon.
Okay, Dr. Antonio. Now we will have the next lecture. And girls, can you?
Okay, thank you, Dr. Antonio. Our second guest speaker today is Dr. Sandra Milak with the lecture entitled Predictive Breeding in the Era of Genome Editing. Dr. Milak has a bachelor degree in agronomy from Federal University of Pelotas, a master degree in genetics and plant breeding from Federal University of Rio Grande do Sul, and is a PhD in genetics and plant breeding from the University of Minnesota. Dr. Milak was a professor of genomics and of genetics and plant breeding at Federal University of Rio Grande do Sul, and also worked at Embrapa, leading molecular breeding efforts in wheat and barley. In 2003, she started working at Pioneer as a a corn breeder, and nowadays she is a member of Qtl Research and Development Leadership Team and the Advisory Committee for the Plant Science Institute of Iowa State University, being responsible for enabling and accelerating breeding and seed product development systems through applied science and technologies. Dr. Milak, thank you so much for agreeing to participate in this IMPB. You may start your screen share, and I would like to remember that our Q&A section is open for questions. Please, everyone, feel free to send your questions, and we will forward the most voted ones to the speaker at the end of the presentation. Thank you, Dr. Milak. Go ahead.
Thank you, Patricia. Can you see me? Can you see my screen?
Yes.
How about, can you hear me okay?
Everything okay.
Perfect, excellent. Well, first of all, thanks for the invitation. This is a community, the plant breeding community and the university community in Brazil that I feel that I belong a little bit, because I've been for a long time, I would say more than half of my career working there, have great still connections and colleagues and friends. And it's very hard to follow Antonio Costa Oliveira, my good friend and good colleague for a very long time. Very well done, Antonio. So today what I want to do is really to try to connect predictive breeding in the area of genome editing. I saw yesterday you guys had excellent talks in the genome editing technology, and today you saw great progress and excellent talk from Antonio in how he's using some of the molecular tools to advance the breeding of cereal crops in the southern Brazil. So I guess the underlying question that we all ask ourselves is genome editing really gonna change the way we do breeding, and is how it will influence predictive breeding? And that's what I'm gonna try to address today to you. But before I do that, what I really would like to do is to really ground us back to why we are doing what we do, why predictive breeding is important and plant breeding is important, why we do all that. And it's really to drive genetic gain. What you see in this picture is advanced trials for corn. You see lots of plots, a view of that. And that still continues to be extremely important in driving genetic gain and testing new products every year. And we do that. And that advancement of products has played a very important role in, in being able to continue to not only drive genetic gain, but also to actually get, solutions to the farmers, better seeds, better genetics that they can really drive better yields in the field. So this genetic gain, and the talk today will be a lot about corn. And that one of the very important things that happen with corn, especially, Exalc has had a huge legacy in both breeding tropical corn and also a lot of genetic studies in corn. Dr. Paterniani, Dr. Venkovsky, and many others that have done an excellent work in this area and have really set up the legacy for, I would say, many of the things that we do, we still do in the tropical corn world. And so heterosis has been probably one of the biggest changes in terms of getting yields in a crop like corn. And you see the picture here on your right. Really, you can, when you compare the hybrid plants or ears in the middle of the pictures versus the inbred lines, which are the parents, you see a huge difference. And the heterosis comes from that complementation. And that drives today, a lot of the gains that we made. But what I'm telling you that is because really this hybrid revolution has really driven huge gains. There was probably a big shift in the gain, in genetic gain for corn. But in reality, there has been a lot of breeding happening in the last several decades, I'll say last century, that has brought corn yields to be where they are. And what you see in this graph today is on your y-axis is bushels per acre, is yield. And on your axis, you see time. And this is 150 years time frame, where for, I would say 50 years, all we did was farmers would pick ears from their fields, and was some open pollinated variety breeding, but it was you could see not much gain happening. It was I'll say in the early 20s, that's when we really start doing inbred development, and then that drove hybrid production in the next 100 years. And you see this curve, and you see this progress. And the question is that, can we continue driving this? I remember when I was going and doing my 101 plant breeding classes with my students, or even when I was a student, as some of you are, the questions we had is like, can we continue driving this? And I think what I want us to think about today is like, can we actually, can gene editing, new technologies, a better understanding of genome, help us to continue pushing this genetic gain even further? And I'll tell you, of course, it was not only genetic gain that made this possible. 50% of this gain came from agronomy practices and management practices. One big one, in corn particular, as you see in this picture, you have corn planted in the 1939s and corn planted in the present days, and you see a lot of differences in planting density, and that has really been a major factor in improving yield in the fields and productivity in the fields. So, but genetic gain has played an enormous role into this. And, to try to understand the magnitude of what that has really brought to reality, I like to see this picture and go over, and this is, higher yields per unit land, and this is the picture US map, where you see here in the squares, the very dark squares are the very high yielding, and you see here the area in corn acres for corn in North America with the 2017 yields. This is the data from 2017. Now, would we actually do a virtual planting using the yields that were actually done that we were able to get in 1926, the levels of that? We have a picture like this. And this is a very dramatic picture, because it means that to get the same level of production of 2017 in corn, would have 1 in every 3 acres of mainland US would have been planted with corn. So we had to plant all the green squares that you see in the right map to be able to get the same production level. So absolutely, genetic gain not only have allowed us to really continue driving productivity per unit of land, but has also allowed us to drive production at a very high level without having to expand the land, with, of course, a great story in sustainability and the impact of breeding and continue driving that for agriculture and I would say even for the ecosystems. So when we look at the global footprint for corn, and here is a picture of corn planted across the globe. You see, the very dark green means, high levels of yield. You can, in this scale, is up to 12 tons per hectare. in this picture. And you see that there are, across the globe, you see lots of, high yielding locations and germplasm, including Brazil. You see lots of very dark green there. But we also see a picture that tells us that this can still play a very important role. So in other words, we still have to continue driving that. And, not only genetic gain, but also how we manage and how we continue improving agriculture as a whole. So very important role, very, very important message about why we do that at the end of the day. Now, I'm talking a lot about corn today, but I want us to ground that what I'm going to talk today, it is about corn, but we have done this for every single crop that we work with. And just to give you a perspective, these are several crops here. Majority of them are hybrid crops. We have soybean and wheat that are still varietal crops. But what we have learned over the years is that this predictive breeding that I'm going to speak a little bit more in the next slides about, it's the principles of that, the fundamentals of that are pretty crop agnostic. Doesn't matter which crop you work, the genomic technology, the experimental design, the digital phenotyping, the prediction methodology, there are a lot of things that are foundational that can be actually learned and transferred across crops and have served us in Corteva to really drive a predictive breeding in all these crops, which we today apply and use. So let's go back to a little bit about predictive breeding and defining what do we mean by predictive breeding, or what we call the modern era of plant breeding. And basically, the very fundamentals of breeding continues to be the same. As a breeder, some of you, and what you do is basically you create new genetic combinations and you create new genetic material, you evaluate and you select for. And the way we do is that is in the form of a shape of a curve where you have a lot of materials that you test at the very early stages of breeding. And then as you go by, you continue selecting and, and eliminating so that you can test and up to the point that you have one or two that you launch. So in the very early stage, it's very expensive actually to do all this by using only phenotype. And the way we have expanded this funnel, or what we call like a wide the funnel, has been by really using genotyping to be able to evaluate that. For us, genotype is much, much cheaper than having to plant a plot, So, and then we can get and evaluate this. Also, in the very early stage of any program, you probably don't even have enough seeds to be able to plant trials with several locations and several reps, or even both. And then so it becomes very important to be able to do this if you want to do in large scale, to be able to do this using genotyping. Now, as you go by in this pipeline, every step, you move from an inbred focus to more like a hybrid focus. You're not trying to just find combining ability, general combining ability, but now you're really starting to see which are the right exactly products that I want to advance later on. Then phenotyping becomes, increasingly important. And at the end, you have to still phenotype and make sure that you evaluate all the new products in trials in the farmer's field. So the reason we have been able to do that, and what we call this predictive breeding, is relying in prediction frameworks, where we're able, using genotype actually, to evaluate and and predict the phenotype without having to phenotype everything. And these prediction frameworks, as you're going to see a little bit today as I walk through it, it's evolving to start also considering what are genotype by environment interactions. Can we start looking to that? Can we start be able to predict products even before we have seen this product in different environments? And can we actually even predict the ability of a particular product, to really perform not only in an environment but also with a particular way of managing that product? So to get to this point in using predictive breeding, it took us some time, right? I mean, but I would say that technology advancements were very, very critical in getting to that. So in today's talk, and for the rest of this talk, what I'd like to walk you through is sort of some of these technology advancements. And it is a lot today about genomic technologies and how that evolved and how that is helping us to get then to the point that we can start asking the questions, can we understand the genes? And can these genes really help us to understand why these crops behave in the way they behave and how they interact with management practices? How will you explain the biology of that? So more and more in how we incorporate that in the way we do predictive breeding. So, you know, I'll say that still, as Antonio mentioned today in his talk, Qtls are still a very important part of that with technology using, you know, just hundreds of markers. We can get to that and identify Qtls and also track Qtls. So it's still a practice we use and many crops are using. But as we move into genome predictions and genome technology became less expensive, I say that we have become more and more advanced in understanding haplotypes and genes. So let's just walk through that a little bit step by step. And let's start with Qtls and the importance of that. And here is one example of bacterial leaf blight in rice, where you can see markers a marker here for XA21 gene. And you can really, you know, see that very easily track this gene using markers and you know, be able to select plants that are resistant versus susceptible. Very interesting, very good application, and this has been a very, you know, and it still is a very important use of genomic technologies to be able to select plants and doing genotyping. Now, what we've learned over time is that doing this for a couple of traits, very important traits, is one thing. Trying to do this for multiple traits at the same time becoming a much harder problem, a much more complicated task. So we learned that, you know, with genome technologies, we could actually, you know, using a training set, estimating marker effects, predict breeding values, and with markers across the genome, we could then start, you know, look at several traits at the same time. And instead of having to track Qtl by Qtl, we could actually do genomic selection for multiple traits. And really this, I would say, genome prediction became sort of a paradigm shift in the way we look at how we could use markers to actually advance in selections in plant breeding. So that has been done. And I would say that it has been a journey for us in terms of getting from that. At the beginning, it was just, as I said, hundreds of markers. But as you can see, you know, what we have done over time was really to start understanding much more the germplasm that we had. And with that, really, we move us into the next, with genome technologies, move us to the next level of exploring this. Let me walk through a little bit how that journey was. The first graph you see in front of you is what we call the universe view of the corn germplasm for Corteva. And why this is a very interesting graph, it shows the evolution of the germplasm and the evolution of heterotic groups, where you can see that everything at the beginning, 100 years ago, they were all the dots in the middle that represent inbred lines. And then over years, with, you know, breeding, several cycles of breeding, we kept pushing and developing more and more inbreds that became different, more and more different, diverse compared to each other. So you can see each one of these dots is an inbred line, and you can see, you know, this is organized from early germplasm to late germplasm on the, you know, and every year you have a new ring into this tree in each one of the sites. So because of the foundation of this germplasm, and because we understand the founders and we understand the genetic relationships very early on, even with hundreds of markers, we start looking how these haplotypes were, and we call identify descent, because you had the genotype information, you have sort of the pedigree information, you could connect those two things and start really designing how this germplasm was evolving. But then this became the foundation to really explore from markers to haplotypes. As we got genome sequencing cheaper and more readily available to do in many more inbred lines, we could then start exploring haplotypes. Let me explain a little bit what we mean by haplotypes and how we look at this. Well, in this graph, what you see is inbred lines from A to H, different inbred lines on the, the horizontal view, and in the vertical view, you see physical position of chromosomes. In the case of corned from chromosome 1 to 10, like, one after the other, all stacked on top of each other. And when you look at this, basically the way we do this is that each inbred is compared to a reference inbred. And in this case, if it matches the same sequence of the reference inbred, it's going to be blue. And if it does not match, it's orange. Okay? So we look at that inbred by inbred, and then you have this picture. And what this picture start revealing us is that, okay, there are some portions of large haplotype matches between the reference and this, in the case of G and H, which are very, very big, large haplotype matches. You can start seeing high diversity where, although, very different from the reference. And the fact that it's orange doesn't mean that it's equal to just, another inbred. It could be, many other colors, right? Coming from many different diversity, different inbreds. And then when you look at another one, sometimes you see places where you have some low levels of diversity, but you do have the, some inbreds that still contain the reference, but some new diversity coming into it. That gave us really a good perspective of understanding the germplasm. This is sort of, so then we said, okay, if we can do that with one or few inbreds, how we actually build that for the entire germplasm? And just to walk you through this haplotype construction is when you look at inbred 1, we start looking, okay, this is, let's call this blue. You can call red, whatever color you want. To say, let's call this blue. And then you said, then we come and look at the second inbred and say, how this compare inbred 2 compared to 1, and you see, okay, there are some, places that are the same, so they are going to be blue. Places that are different, the second inbred, now we're going to call red. Then we compare to the third one, and then we look at, there are, inbred 3 has blue, red, but also has yellow that do not match with either one of the previous inbreds. And then inbred 4, for instance, is completely different, is green, does not match with anything that you have, which would be a very specific case for a tropical germplasm compared to, temperate germplasm, very different in haplotype constitution and sequence level. So when you start building that one by one, what we've done over the years was to say, can we actually build a library and understand really all the inbreds that we have? And here is a sample of that, where you look at this picture, you see on the horizontal line here, it's on top is our inbred lines, one by one, inbred lines. And in the vertical is a physical position, again, the same as I presented before. And you have two sets, set 1 and set 2 here. And you can start having, this is a sample of the germplasm across the globe. And you can start looking this and you see, I can see reds across in some places, but I can see also pools that have almost no red. So it means that the level of diversity and the matching of this sequence level between this red haplotype with this germplasm is 0, and so on and so forth. So with that, you start really understanding a little bit more at the haplotype level. This was a very important step for us to say, now that we have this, where we go next, right? How can we utilize this to really drive the next level understanding? And I'll say that that's where the genome editing, we sort of start pushing also this envelope, I would say, in terms of technologies, because in order for you to do genome editing, you have to have high quality crop genomes. You know, you have to have, because you cannot really do this in an effective way if you cannot, you know, design guides for CRISPR with, you know, you gotta have sequence and good level of sequence. So what, with the genome editing era, what, you know, many groups have done was to go back and say, you know, do I have that sequence and how do I get it? And so we had multiple reference genomes being built, what we call the pen genomes, where, you know, the very high quality crop genomes. And with the genome technology evolving, I think that has become a much easier, you know, task in the terms of like an affordable task, I would say. So with all this being built, then we start asking the questions, well, now that we have this, can we actually build and impute this haplotype level that we had about the 1 centimorgan level to actually real genomes, full genomes, and even to genes to understand that, right? And I'll say that we are in that journey of being able to do that. Today we can actually do imputations of the genome and really get to almost, you know, a sequence level genome for almost every single inbred that we, you know, we have information for, you know, a skin sequence level of sequence, I would say. And then with that, because you have that information, then you can start asking yourself, what do I do next? So basically, we have used this information. Here is one example where we had a Qtl map, and using this haplotype and this sort of pan genome level of understanding of this germplasm, we could start finding, you know, go to a region where we know where the Qtl is, actually go to a very narrow region of that haplotype and understand how many candidate genes we have there. And okay, if we, in this case, we had, you know, a handful of candidate genes, and then you can go and use genome editing, CRISPR technology, to knock those genes and do a very quick phenotype test to say, can this really disrupt this gene, and do I get the phenotype that I expect? And here is one example where out of this, you know, handful of genes, we identify one that was the one expected for the male sterility in sorghum. So confirming that that was the right gene. So now, what took us a long time to go from Qtl to the gene, because we had to do fine mapping, a lot of fine mapping, we had to do all these steps, and it would take years to even develop these populations. Now you are in a point that you can really take, once you identify this, if you can get that resolution of haplotype, understanding of your germplasm, and you get to the resolution of understanding the gene sequence of that germplasm, you can use the genome editing technology to really drive this, to get to the genes. So why does this matter, right? That's probably the question. Yeah, oh, it's great to find the genes. So what do I do with that? And how this helps me to advance predictive breeding? These are the questions that we ask ourselves. And I'm not gonna say that we have all the answers, but I can say that we have already some insights in how this may help us. And here is one way that we see that, right? of getting. So just, you know, again, this is sort of the evolution of that. And I walk you through this. And, you know, it goes from 100 SNPs to thousands of SNPs or millions of SNPs to get haplotypes and full gene sequence. And that has been the journey of it, right? So let's see how this helps us. And here is one particular insight that, so as you know, there has been a lot of crop growth models built by, you know, plant physiologists over years. And those have been very hard to do and hard to, you know, get the information for. What you see in this particular slide here, it's actually the pick from a paper from Frank Technot, one of our, you know, colleagues in Corteva. And what he did here was to use an association of these crop growth models with whole genome prediction to try to predict products in test whether we could really predict products in a better way. And in the graphs that you have here, you can see in the y-axis, you can see the predictive grain yield. And in the x-axis, you can see the observed grain yield. So is the, you know, what you see in the field and what you're predicting, do they match? So if you get like a straight line, it means that, you know, prediction is very good. If you don't, it means that the prediction is not so good. So, and these are 2012 data and 2013 data, 2012 being a year with a lot of drought in US. So, and in 2013 being a good year for yields. So as you can see, you know, using the crop growth model and associated with whole genome prediction versus the standard genome predictions, you have a lot more accuracy to predict performance under drought stress. And I think, you know, why this would, the explanation for this is that you're taking a lot with the crop growth models and using whole genome predictions to expand this to the entire, you know, set of populations here. You can really start getting some biological models being incorporated with whole genome predictions. And with that, you start going from just, you know, the predictions and the genes, but understanding also the components of that and how that helps you to explain performance. So what we believe, and just one of the insights, is that in this journey of going from Qtls to actually genes, and how the technology has helped us to advance in that journey, I think what we are gonna get in genome editing will be another tool that will help us to get there, is a much better of understanding of the biological models. And this will have implications in being able to predict performance and hopefully to start solving some of the challenges that we have had in predicting genotype by environment and by management interactions. I think, you know, when I look at the sort of the frontiers of plant breeding, I think this is one of the frontiers. The reason why it is because we have had very good and excellent, you know, progress in predicting using genotype. You know, technology advances have put us in a place today that the accuracy of doing predictions is as good, or sometimes, or most of the times better than actually measuring something directly in the field. You're just better. And so there's no doubt about that anymore. I think that, you know, is a frontier that we already crossed. You know, it's not future anymore, it's present. But now we are starting asking, where do we go from here? And I think one of the areas that I see in plant breeding that we haven't been able to have a good handle of, because it's a very complex area, is how products and germplasm interact with environment, especially year to year, because it's very hard to predict what happens. But with more environmental data and in being able to really connect data in different levels of complexity, and also being able to understand more the biological models, because now we understand better the genes and, you know, and understand that, I think will really help us to go into that. Also, I think another area is like how we understand the complexity of, epistasis and other types of gene interactions that today the models we have for prediction, they probably don't take that into account. They're highly based on additivity by additivity and additive models. So how we start looking some more complex type of interactions as well. So that is sort of, what I, want to talk to you today. But I want to leave you with a story, which I think is sort of a little bit my personal story into this. And I thought it was, you could be asking the question, okay, this is great, it's corn, a lot of investment in corn, you can do this in corn. How about the other crops? How about the very complex crops like oats and like, wheat, like as Antonio mentioned, what we do with those crops? Can they benefit from this? And I'll say that these are, especially oat, very complex genome. I think it's at least 5 times bigger genome than corn. So that is just the first, one of the first things. But there is still one thing that has happened very recently, and this is a partnership between Corteva and PepsiCo, was the release of the full oat genome. This was a project that, really, we believe that this becomes the foundation of understanding the genetic basis for many important traits. And this genome is fully available, public. You can click in this link and get to the genome. It's all, it's out there. You all can use it and explore in your research. And the reason why this is a personal story is that when I started my PhD many years ago, 30 years ago, my PhD thesis was with oats. And my project was actually funded by Quaker Oats, which now is part of PepsiCo. So, that was completely funded by that. And my work was about mapping Qtls for dwarfing genes in oats. And by then the technology, so I can tell you the story from there to here, because I've probably seen that in a crop that is more likely done in the public sector. But I've seen the evolution of that. And when I did my PhD, we had 45 linkage groups. And these are, oat has 21 pair of chromosomes. We had twice as many linkage groups because we couldn't connect any of these things. The maps were very, difficult to do, a lot of complexity in the genome, not many markers. The maps were still very not full maps. An exaploid crop, very complex. And it's interesting, when this came up a couple of months ago, one of my colleagues told me, send me a message, say, Sandra, what would you ever dream in 30 years ago that you'll be able to get a full genome? And I'll tell you, the quality of this genome is phenomenal. Some of these chromosomes are pretty much one sequence, one entire long sequence. So it's just amazing to see that we got to this point. And why I'm telling you the story, because question I ask for most of you that are young people starting your careers, that what the next 30 years are going to come and do for us, right? So what I've seen in my 30 years of working with plant breeding is that the pace of technology has been accelerating faster and faster. And I think because of that, I think we will probably see some very fundamental changes in the way we do and we breeding in the next 30 years. And you're going to be living that life and you're going to be able to participate into that. So what I say, don't think that this is only a corn story. I think this is a story that we will be talking about in the next many years in front of us, in many, many more crops. And I think you guys are going to drive that. So I'm glad to see that. So with that, I'll take any questions, and I want to thank you again for the opportunity to be with you today.
Okay, thank you, Sandra, very much for your great lecture. We are very happy with that. So we have some questions now. We have a big one here in Q&A. Do you think that the haplotype analysis will overcome the traditional SNP by SNP analysis? Is there a way to quantify the percentage of coincidence we have between genotype haplotypes? What is the ideal haplotypes slices size in maize?
Let me just close here. Try to get back here. Okay. The answer to that question is yes. For us, haplotype analysis has already replaced SNP by SNP. So we're no longer doing SNP by SNP analysis. It is a whole genome view of it, and is really based on, you know, haplotypes and how we look at the germplasm, how we make decisions about the germplasm, the crosses we make. All that is based on haplotypes today, and yes. So that is the first part of the question. Let's see if I can see the other parts of the question. Maybe. What is the ideal haplotype size in maize? So we have a 1 centimorgan type of approach for that, and I think it's being, for us, I think it highly depends on the germplasm that you're looking, because we look at very global germplasm and tropical germplasm with temperate germplasm. I think 1 centimorgan has given, for breeding purpose and for predicting breeding purpose, have given us enough view of what we need to make the decisions we make. So that's probably enough. But I would say that haplotype sizes depends on the complexity of the genome. And also the resolution of that depends on how well you can actually sequence this gene, you know, your lines. So more you get, you know, more defined and refined haplotypes you can actually get. But also you don't want to go too small, and neither you want to, you know, you want to go as small as possible to represent your germplasm, because the other thing is that, you know, it also has to match, you know, the linkage disequilibrium of your germplasm. If you might have some big blocks that are what they are, so breaking apart in smaller might not just make any sense, because that's the way the germplasm is organized. So I think it's a fully loaded question, I would say. There are many angles of looking at this, but for specifically corn, for us, 1 centimorgan haplotypes is what we are using now, and it's being effective in driving all the breeding decisions that we need.
Okay, thank you. We have one more. Is there a move towards using epigenetic and post-transcriptional data into breeding? Or will DNA remain the only player?
We are not using breeding at this point, but we have done a lot of studies and identified different things beyond these SNPs that really explain, you know, variability. One of them is really our structural variation, you know, presence and absence of segments. We have some very important ones that really, you know, could further explain variability and could even further enhance predictive breeding. So the answer to the specific of the epigenomic aspect, we understand that that's real, that is important. I don't think we are at the point to use it at this point. And whether it's going to have an impact in predictive breeding, the level we have, the accuracy that we have, I would say might increase a little bit, but I think it's still the bottlenecks for moving accuracies in predictive breeding are not going to come from this area. It will likely come from refining our ability to measure phenotype. which is another big area of, you know, of research today, the digital phenotyping area. That's probably has become the next big bottleneck for everyone working with predictive breeding.
Okay, we have one more from Mario Sigris. What or how should be considered regarding the impact of a edited germplasm in prediction models?
And I'm not sure if I fully understood the question. I don't think that I don't know if I understand the question, but you know, I think that gene editing at this point, what we'll do, it will have an indirect effect into that, ability to improve predictions by when we get to do this to many more genes and understand the biology genes, as we advance that, I think there will be an increased understanding of the sort of biological models and this whole genome prediction, crop growth models area. I think gene editing can play an important role there to help us to understand better candidate genes and understand those biological models. And that, I think, will have an impact in really, in my opinion, in moving our prediction models to the next generation of it, where we can use that framework to understand how, you know, different germplasm behaves in different environments. You know, once you understand the biology of it and you understand those interactions and how they happen and how they are triggered, you can start trying to understand what is the potential, you know, if you have a very drought location and a location that, you know, has a high probability of becoming a drought, you know, location every year, you know that that is the type of germplasm that is going to perform, because you know that these are the type of, you know, genes and biology that is underlying that set of germplasm. I think that's sort of the mind set for thinking about that. In the current ways where we do predictions, I don't think we will change anything, but we will change how we incorporate this in the future. I hope I addressed the question. I don't know if I understood the question correctly. I hope I addressed the question. If not, please ask again and I'll give another try.
Mario said that he understood and he thanks for it. I have one question too, Sandra, if I can.
Yeah.
What are the major challenges do you see in application of predictive breeding in tropical environments, principally with the climate changes?
For me, the using predictive breeding, the challenges are more in building that infrastructure that allows you actually to get to start using predictive breeding, right? In tropical environments, you'd have to have, you know, training set. First, you have to have tropical germplasm with high level of, you know, genotyping and phenotyping in the environments that matter to that tropical germplasm. So you can create, you know, training sets that can then be used to predict your germplasm, right? So that foundation for me is probably the biggest challenge, to be able to build that with data, with good data, phenotype data that can really be, you know, help be clean enough to be able to predict that germplasm, right? So I think that's sort of you know, one of the challenges. Climate change, I think what happens is goes back to a little bit of what I mentioned before with the other question, is what if one thing that climate change is going to do is just to create more interactions, new interactions that we may have not measured before, and that we may not really know will be, for instance, new diseases that, you know, we never really understood. So we don't have a way to predict, right? Because we just have not been able to measure or understand. And I think, you know, if we are able to actually keep up with the phenotyping and be able to really do that well, I think predictions can really help us, because the genotyping part, once you have it, then it's just a matter of being able to really inform those models to predict the new traits that are coming in the new environments that are coming. But again, the challenge for me is more to be able to do that, to really do a very good job of measuring those phenotypes in those different environments that are going to be starting to show up as, you know, temperatures rises in different places. You know, you start seeing diseases moving to different places that they never been, moving towards in the south hemisphere, moving from from more of the warm regions of the high and low lands in, like in Brazil as one example, to the, you know, to the southern part of Brazil. New diseases are moving. We already see that happening. And that germplasm has never been exposed to those diseases. So how we make sure that we understand, measure those phenotypes, understand and characterize that, and getting ahead of that may mean that we may have to start moving germplasm to, you know, different regions to be able to characterize, so that we are ready when the problems come. But that's going to be the challenge, is be able to do that. Not predicting. The prediction models work, and they can, once you have, you know, that done, it'll be easy to get there.
Okay, thanks, Dr. Sandra. We have a question from Dr. Antonio. Between those heterotic groups, what would be the role contribution of different alleles and missing genes in each other? I mean, different alleles for absent genes in each group.
That's a great question, Antonio. We actually have done a study where we look at presence and absence of alleles in complementary heterologous groups to see, is that the way we are getting heterosis, because this, you know, missing genes now are present in the other side, so they compensate each other. I think maybe it's probably where we are going with this question. And we have actually identified that there are some regions that is the case, and that explains likely a portion of why heterosis is happening. So yeah, so it's real, and we have identified to some degree that it's probably one of the causes of those that is driving heterosis, presence and absence. And now we are, you know, and how we incorporate this in the prediction models and capture that. I think that's where we are at.
Okay, let me see. How genome editing for one or few in an already highly domesticated could still be aided by predictive breeding?
Can you repeat the question, because I couldn't quite understand the question.
Okay, sorry. It's on the chat if you want to see.
Is it in the chat?
Yes. How genome editing for one or few in an already highly domesticated could still be aided by predictive breeding?
So see if I can understand the question. I'll try. I'll give a shot here. So yeah. So I think the problem is not editing this in a, you know, not non-domesticated crop. I think the problem is, if I understood correctly, so if I don't think we'll change predictive breeding as well. They might complement predictive breeding, the ability to be able to edit a gene in a allelic germplasm with a potential allele that is from, that has been identified in the exotic, if that is what I understood the question was. I think what that will do is just will complement the ability to move variability into elite germplasm, because now you have a very high level of elite germplasm that you've been driving it with predictive breeding, and you have this new allele that you can go and just change that precisely. So without having to bring a lot of other, baggage from the, the sort of more exotic germplasm into the elite germplasm. If you can really precisely just change that in the elite germplasm, that will help to complement predictive breeding in the sense that now you can just keep, moving in without disrupting the germplasm. And I think that's going to be one of the things. How that is going to impact highly, I think, depends on whether genome editing will be or not be regulated. That's one thing that we're still trying to understand better. So if we get to an ideal world where we can do gene editing in any germplasm, and that is considered GMO or regulated or whatever, I think then I think it just opens other avenues for us to go beyond that only one by one allele editing. One thing, for instance, and one of the things that we discuss internally in Corteva is that would we get to the point that we could just use gene editing with no regulation? Could we actually start identifying deleterious alleles in our germplasm and just delete those genes, right? And do that in, at the same time in the germplasm for many genes, right? That I think probably can help us to even move the needle further in that direction. But again, I think that, gene editing as a tool for predictive breeding today is more like a discovery tool to understand, not and a complementary tool for the germplasm the way it is. I hope I addressed the question, if I understood correctly.
I think that was the question. Thank you. Another one in the Q&A. In your opinion, what would bring more genetic gain to the crops? Gene editing or genomic selection, or both? Gene editing will replace the actual OGM techniques?
It's a fully loaded question. Let me take piece by piece, okay? Because there are lots of questions within this one question here. Let's get to the first one. Will be more genetic gain to the crops? I don't think there'll be more genetic gain. I think gene editing will play a very important role in actually fixing specific problems in the germplasm. But again, and that I think at this point that's the way I see it. So unless we can actually do gene editing in multiple regions of the genome at the same time, in very early stage, so that whatever germplasm we get is already edited for like maybe 100 genes or something like that, I think unless we get to that point, I don't think genome editing is going to play a role in bigger than, than what we already do with breeding. It will be though fundamental to really change specific problems and target specific situations. And I think that becomes extremely important depending on the germplasm you're using, right? So, I mean, if you ask the question in a different way, you say, would you be, if you have to use a very exotic germplasm to incorporate a particular disease, can you make more gain by doing this by breeding versus by gene editing? I'll say you can make more gain by doing this by gene editing because you're not disrupting the very elite germplasm. That's one way to answer the question, right? But I don't think there is, I think gene editing would be more to complement than just to replace predictions and breeding in that sense. That's the first part of the question. The second part of the question is, will gene editing replace GMOs? So I think gene editing is effective if the alleles that you're trying to change are present in your germplasm, right? That's what genome editing is about. You take, you can either delete an allele, you can move an allele, or you can stack, but these are all genes that are present within the species. GMOs are beyond that, right? I mean, you gotta bring genes from different species in GMOs, right? And I think there are some genes that we haven't, unless we find this in plant species, in their own species, but like, I'll give an example of insect control is one of them, right? The BT genes and some of the other genes that have been identified. Those do not exist in plants. So I don't think gene editing will be able to replace that part. We still have to use GMO techniques to bring some species exotic genes to be able to bring to plants. I think that's still going to be a very important role of GMOs. So I don't think it's either or. I think there are some applications of GMOs that you may be able to actually replace with gene editing. I mean, if the gene is already, like, I'll give an example of herbicide tolerance. If the gene exists in the species and you can go and knock the gene out or do something about the gene in your own species without having to bring this from another one, that might be one way that you can replace that. But I don't think it's just either or. I think that we're still going to need both of these tools to be able to fight the problems we have, especially if, we're going back to the climate change comment or question, where, you imagine more diseases, more insect problems, more of those things coming. You have to use all the tools in your toolbox to be able to really cope with some of those challenges. And especially if you are in the tropical, part of the globe. So.
Okay, thank you, Sandra. Thank you, Dr. Milak, for sharing with us all your experience and for all the clarification about your excellent lecture. Now we will have the third research presentation session with the participation of Marcos Antônio de Godoy Filho. He completed his master's degree at the Luiz de Queiroz College of Agriculture and is currently working at Neoprospecta. He is presenting his work entitled De Novo Assembly of Contrasting Soybean Genomes for the Study of Resistance to the Stink Bug Complex. Marcos, thank you for your participation. Você pode compartilhar sua tela e escolher se quer fazer a apresentação em português ou em inglês. The Q&A is open for any questions.
Boa tarde a todos. Muito obrigado pela presença de vocês aqui. Só compartilhar, colocar no modo tela cheia. Pronto. Vocês estão vendo a tela?
Estamos, Marcos. Tudo ok.
Ok. Bom, vou apresentar meu trabalho, que é intitulado Montagem de Novo de Genomas Contrastantes para o Estudo da Resistência ao Complexo de Percevejos. Esse trabalho foi feito em colaboração com a Universidade de Illinois e também com a USDA, com o Steve e o João Paulo. Bom, a soja é a principal commodity brasileira. Há vários anos ela é o principal produto de exportação brasileiro. E só em 2019 a soja e seus derivados geraram mais de 30 bilhões em exportações. Entretanto, para a gente conseguir lograr grandes produtividades, nós temos também grandes desafios, como o complexo de percevejos, que é formado principalmente por 3 percevejos, o Piosodoros, o Unisara e o Euquistos. Bom, e o controle desses percevejos é realizado, então, geralmente com inseticidas, misturas de piretroides e neonicotinoides, que apresentam baixa eficiência e também um alto custo aos produtores. Além disso, milhões de pessoas acabam sofrendo de intoxicações por inseticidas todos os anos. E uma alternativa do uso de inseticidas é, então, o uso de genótipos resistentes. Podem apresentar antibiose e antigenose, que seria a mortalidade dos insetos, ou então a não preferência alimentar deles por certa variedade, e também a tolerância, que é dada por diversos caracteres agronômicos. Entretanto, nós temos algumas, aqui no slide tem uma listinha com algumas variedades resistentes, mas elas apresentam geralmente baixa adaptabilidade, conseguem até alguma produtividade boa, mas só apenas em regiões muito específicas. E como acontece a resistência? Nós temos uma parte química, que são os flavonoides, que vão levar a antibiose e antigenose, e também alguns caracteres agronômicos, como menor período de enchimento de grãos, um menor tamanho de sementes e, consequente, um maior número delas, uma reposição de vagens danificadas e um alto número de vagens nessas plantas resistentes, e uma menor retenção foliar. Os percevejos, além dos danos nas sementes, causam também a retenção foliar, que é quando a soja se mantém verde durante todo o seu ciclo. Bom, e quais foram os objetivos do nosso trabalho? Primeiramente, nós obtivemos os genomas da IAC100, que é um genótipo resistente ao complexo de percevejos, e a Codetec 215, que é suscetível. E diferente de dados de GBS ou então de chip, que as pessoas estão mais acostumadas, esse tipo de dado de alta profundidade de sequenciamento permite que a gente consiga identificar outros tipos de polimorfismo, outros tipos de variantes, como, por exemplo, indels e variantes estruturais, como presença e ausência. Então, outro objetivo foi identificar essas variantes em regiões e também alguns genes candidatos em regiões com alguns Qtls reportados para resistência, e identificar algumas variantes únicas provindas da IAC100, presentes numa população de rios que nós temos no laboratório, que é uma população originada do cruzamento desses dois genótipos. Para o sequenciamento dos genomas, foi utilizada a tecnologia 10X da Chromium. Basicamente, nós vamos ter grandes moléculas de DNA que vão se ligar a bits e receber um barcode único. Teremos o processamento ali dessas sequências e o sequenciamento da Illumina padrão, short reads. Então, teremos um barcode nessa short reads que pode ser ligada de volta a essa grande molécula. É o que nós chamamos de linked reads. Elas simulam ali long reads. Para a montagem dos genomas, então usamos o software Supernova, que é baseado em grafos de Brain, que conta com a ajuda ali das linked reads, né? Para a ordenação do nosso genoma, nós usamos o Ragu, para correção o Reaper e uma nova reordenação com o software Ragu. Para anotação, nós usamos o RepeatMaker e o Augustus. Para avaliação da qualidade dos genomas, nós usamos o Busco com o banco de dados das embriófitas. Esse banco de dados tem vários genes ortólogos únicos e a presença deles nas nossas montagens significa que a nossa montagem está bem completa, vamos dizer assim. Então, quanto mais genes, melhor. Para uma análise de sintenia com a Willis 82, fizemos os alinhamentos entre os cromossomos das nossas montagens contra os cromossomos da Willis 82, usando o NUCMER, e alguns scripts em PER, para avaliar, por exemplo, Contiguen 50, que é uma das principais métricas de qualidade de genomas, que mostra para a gente quão contínuo vai estar nosso genoma. Bom, a partir dos dados de sequenciamentos, ainda fizemos uma chamada de variantes, de SNPs e indels, a partir do pipeline do GATK, e observando e também usando os alinhamentos gerados com essas reads, conseguimos identificar, manualmente, algumas variantes estruturais perto de QTLs selecionados ali do trabalho da Muller de 2017. Selecionamos 3 QTLs. Bom, a população de SNP, de rios que nós temos, são 236 rios. Nós temos dados de GBS delas, usamos elas para a chamada de SNPs, e selecionamos as 25 mais resistentes dessa população, e selecionamos os SNPs compartilhados por todas essas 25 que foram originadas da IAC. Usamos os dados de alta profundidade para fazer essa comparação ali. Bom, a nossa montagem final dos genomas gerou scaffold 250 muito bons, superiores até a diversas outros genomas de soja disponíveis na literatura, como o Willis 82, o Anhan 13. Lembrando apenas que esses genótipos, eles usaram diversas técnicas de sequenciamento, long reads, short reads e até mesmo métodos citogenéticos. Também conseguimos um número de scaffolds muito bom, reduzido. Lembrando que a gente sempre quer o número de scaffolds o menor possível. O conteúdo de GC também dentro do esperado para genomas de soja. E o tamanho da montagem também muito dentro do esperado para genomas de soja. Bom, na análise de ortologia, de genes ortólogos únicos para avaliação da qualidade, nós encontramos 97% dos genes nas nossas montagens, mostrando que nosso genoma está bem completo. E também com parecido com o Willis 82 na questão de cópias duplas e cópias simples. Para análise da sintenia, nós temos à esquerda o cromossomo 1 da IAC contra o 1 da Willis 82 e à direita o cromossomo 1 da CD contra o Willis 82. Podemos ver aqui pelo alinhamento em vermelho, aqui cruzando a diagonal, que eles se alinham praticamente completamente, sem grandes pontos disruptivos, mostrando ali a integridade dele, e todos os cromossomos das nossas montagens apresentam esse mesmo padrão. A anotação gerou aproximadamente 44% de elementos de transposição, o 82 tem mais ou menos 43%, e o número de genes também esperado, de acordo com a literatura, aproximadamente 58 mil genes, 59 mil genes. Bom, chegamos na parte das variantes, a partir dos dados do genoma. Conseguimos encontrar mais de 290 mil SNPs para a EAC100, exclusivos da EAC100, e mais 335 mil indels exclusivos da EAC100, quando comparados com a CD215. Essa variação entre eles, a diversidade genética entre eles, usando esses dados, mostra basicamente a diferença deles, e dentro dessa diferença, com certeza, nós temos variantes que são responsivas ao complexo de percevejos, que são envolvidas com a resistência e que podem ser utilizados em futuros estudos. Bom, a partir dos dados de alta profundidade, nós ainda buscamos, né, como eu comentei, variantes estruturais, e esse tipo de variante, apesar de não ser muito utilizado, vem cada vez se mostrando mais importante para diversas características. Aqui nós temos um exemplo de uma variante estrutural de mais de 2.500 pares de base, e aqui nós temos uma lista com algumas posições das variantes e os seus respectivos cromossomos, sempre lembrando que elas estão ali, nós buscamos perto de Qtls, né? E próximo dessas variantes também encontramos alguns genes muito interessantes, genes candidatos, como alguns genes envolvidos nas rotas dos flavonoides, né, conhecidos por darem a antigenose e a antibiose, e também um gene muito interessante, o GASA11, que é de uma família de genes responsivos à giberelina, ativa em regiões de desenvolvimento, como as sementes, e nós sabemos que a giberelina é um indutor de maior número de vagens na soja, que é exatamente uma das características de resistência ao complexo de percevejos. Bom, a partir dos SNPs, das 25 rios mais resistentes da nossa população, também pudemos, e compartilhados apenas com a EAC100, conseguimos então identificar diversos haplótipos aqui, presentes nessas rios mais resistentes, e muitos desses haplótipos batem com Qtl já relatados na literatura para resistência a percevejos do trabalho da Moller. Bom, chegamos às conclusões do trabalho. O uso das linked reads, então, podem ser uma ótima alternativa para obter genomas a um baixo custo, e além disso, estamos provendo 2 genomas de soja brasileiros com os melhores scaffolds N50 reportados até o momento. E ter esses 2 genomas representa provavelmente uma redução do que nós chamamos de viés de referência para estudos de resistência ao complexo de percevejos, melhorando assim então a chamada de SNPs, por exemplo, e consequentemente a qualidade de estudos como seleção genômica, JUOS, mapeamento de Qtls e até mesmo estimativas de fold change para análises de expressão diferencial com RNA-seq. As variantes que nós encontramos nesse trabalho, muitas delas podem estar associadas com a resistência e serão utilizadas em futuros estudos, como, por exemplo, o mapeamento fino das regiões de Qtl já mapeados. E esses resultados também abrem as portas para diversos outros estudos e aplicações, como modificações genéticas usando CRISPR-Cas9 em alguns genes candidatos. As referências. E meu muito obrigado a todos os parceiros, ao laboratório e ao GVenc. Fico aberto a perguntas.
Ok, Marcos, muito obrigada pela sua apresentação. Eu gostaria de saber se você tem uma ideia de como que funcionaria a validação com dados de transcritos a partir dos resultados que você teve.
A partir dos dados de transcrito, a validação do que exatamente? Desculpa.
A validação das regiões que você encontrou e dos genes candidatos.
Certo. Nós teríamos, então, uma lista, né, de genes candidatos a partir do transcriptoma, das expressões diferenciais com RNA. Nós podemos comparar se os genes diferencialmente expressos estão são de algum gene dentro dos Qtls, por exemplo. Se esses genes estão próximos de Qtls candidatos, de Qtls já mapeados, ou se é até algum gene dessa pequena lista que eu mostrei para vocês.
Ok. More questions, someone? Eu gostaria, então, de agradecer mais uma vez, Marcos, a você ter apresentado e ter submetido o seu trabalho.
Eu que
Em
agradeço.
nome do GVEM, que todos nós.
Muito obrigado.
E, então, nós vamos dar sequência agora. Now we will have a poster session with research posters from participants who submitted abstracts. Their email contact will be available, and all participants may send questions and comments directly to the author. We will be back at 4:40 p.m. with the lecture of Dr. Lázaro Pérez.